Okay, so we start again, and I'm very happy to announce Til Ole Bergmann as next speaker. Til is Associate Professor of Neurostimulation and Deputy Head at the Neuroimaging Center at the Johannes Gutenberg University Medical Center and Leipzig Institute of Resilient Research in Mainz. He obtained his PhD at the University of Kiel. Then he also traveled, working at Radboud University and University of Tübingen, and arriving in Mainz in 2018. His research is focused on the development and application of multimodal non-invasive neurostimulation and neuroimaging techniques. He combines neuronavigated transcranial brain stimulation techniques such as TMS, transcranial electrical stimulation and transcranial ultrasound stimulation with concurrent functional magnetic imaging and electrode and magnetoencephalography, EEG and MEG. So he uses a lot of combinations of techniques, and today he's going to talk about combining TMS with EEG and fMRI for state-dependent stimulation and causal network interrogation. Okay, thank you.
Okay, thank you very much for the introduction and for having me here. So, I'll set my clock. So I'm going to mainly talk about TMS, or exclusively today, and its combination with EEG and fMRI. So the talk will be split in two parts, basically, for these two recording modalities. I would start with a brief conceptual thing about cause inference, basically picking up also the topic that Gesa has already introduced in her talk. So typically, when we talk about brain stimulation studies in cognitive neuroscience, you read sentences like, here we use brain stimulation technique X to stimulate region Y, and this is going to change cognition or behavior, function Z, whatever. And it sounds very simple, but typically there are a lot of more implicit assumptions behind that, right? Because it means you might be applying TS or TMS to induce a certain E field in the target region to then change local neural activity in that region, and then either by modulation of that region or remote network regions, you then have an effect of on a certain neuron processing and a cognitive function of interest, which you then assess again via a certain behavioral task. So all of these cause-effect links have to be valid to actually make the conclusion eventually, right? And there are a lot of possible alternative explanations and a lot of confounders. For example, you might have an E field in non-target regions, right? If the field is larger or differently distributed than you would expect, you change neural activity in non-target regions, which can explain your effects or actually cancel them. If possibly remote effects that were not intended interacting with the actually intended effects, change cognitive functions like attention or working memory, which have then secondary effects on your task performance, etc. It's also possible that you effects of not stimulating the brain at all transcranially, but actually from peripheral co-stimulation confounds. Closer? Yeah. I think it worked yesterday, so I just tried to stand here. For example, the TMS click sound, the tingling sensation of electric stimulation in the skin, etc., can also activate sensory regions, which then activate brain regions involved in the task, for example. So a lot of confounders here, and then task demands, brain state, etc., also can affect each of these steps here. The problem is that many of these variables are latent and not directly observable. E fields we can simulate, it's more difficult to measure them directly. Neural activity we can at least measure with neuroimaging techniques or electrophysiology. Cognitive processes we can model, and motor responses then during the behavior task, well, we can use proper psychophysics to get most of the information out of it. And I'm actually, so this is not only true for electric stimulation, but also for focused ultrasound. Even though it can be more focal, you still have to worry about off-target stimulation, right? Especially when it's a maybe elongated beam, or as we've heard, due to bone aberration, you're stimulating slightly off-target. If you're adding for small structures in the brainstem or in the basal ganglia, just being a little bit off half a centimeter can make a huge difference. And it's, well, not about electric fields, but about the acoustic pressure. And you can also actually hear the stimulation if you use certain pulse repetition frequencies, which is, again, a confound. So basically introducing then the imaging work I'm going to show, we can use neuroimaging either following offline TMS, for example, or concurrent with online TMS for proof of neural target engagement, to show that we've actually modulated neural activity in the region that we were interested in. So we have a lot of different imaging techniques available here, shown according to the temporal and spatial resolution and precision. And we can combine, for example, EEG and fMRI to get the best of both worlds. But, well, as we all know, this is why we're here. We need brain stimulation to get the causal impact and causal manipulation of brain activity. And we have all these different techniques that have a similar spatial and temporal resolution. And we can combine them as well with, for example, EEG, MEG, fMRI. And I'm mainly going to talk about TMS fMRI and TMS EEG today. Starting with TMS fMRI. Why would you do it? Because so I'm skipping all the technical stuff here. I have like another 50 slides in the appendix if you're interested, right? But it's so it is difficult to set up. There are a lot of technical issues. So why might it be still worth doing it? So what are the insights to be gained? One thing is, as I mentioned, proof of neural target engagement. Not only at the site of stimulation, but in the entire network that you are stimulating. We know TMS has network effects, right? Because the brain is connected. That's not a limitation of the technique. That's a feature of the brain. So to show that there's actually activations in the remote regions that we might even want to target indirectly, or at the stimulation site, imaging can help you with whole brain coverage to see where you get a bold response to your stimulation. We can systematically investigate certain call orientations, for example, of stimulation intensities, to see which one actually provides the strongest target engagement, to personalize stimulation empirically. We can also investigate state dependency, and there's a lot of TMS fMRI evidence for state dependency that especially also the remote activations of the connected networks depend a lot on what the brain is doing at that time, whether you're processing stimuli, where engaged in a task, where your attention is, etc. And this also helps us to interpret our results, also our behavioral results when we do TMS doing a cognitive task, for example, because we typically assume when we're stimulating a certain brain region and then we get drops in task performance that this brain region is causally involved, right? But we know we're not stimulating that brain region alone, we're stimulating a network. And maybe another connected brain region actually is more predictive of your behavioral changes. And maybe you also have co-stimulation confounds that you have ignored, but they will show up again in the fMRI, and the auditory cortex will lit up because you have the click sound of the TMS, etc. So it can help you to make sense of the neural substrate that actually produces the neural effects that you're observing. So I want to show two studies that we're conducting in my lab at the moment. One is about proof of target engagement at the stimulation site in the primary motor cortex, and the other one about targeting deep brain structures like the ventromedial prefrontal cortex or the subgenual anterior cingulate cortex. So starting with the first one, now we're basically looking at the bold response directly at the stimulation site. And that has been proven to be notoriously difficult because in a lot of TMS fMRI studies, we don't really see effects under the stimulation call, which can have a lot of different reasons. But it has been shown even in the early studies already that you get when stimulating the motor cortex, an entire motor network activation, right? Even at subthreshold intensities. but only at superthreshold intensities you start getting actual activation of the primary motor cortex, or motor and somatic sensory, so sensory motor cortex. And people have tried to tell this apart using also sensory stimulation, because the problem is when you have superthreshold stimulation of your motor cortex, what you get are muscle twitches, right? And these muscle twitches in your hand, the MEP, that is going to produce reafferent feedback. So you get afferent feedback into your somatic sensory and also in the tightly coupled motor cortex. So maybe the reason why we see only activation in the sensory motor cortex for high stimulation intensities is because this is when we get the muscle twitches. Another reason why sometimes one might not see any activation below the coil is that the coil basically reduces SNR, so the TMS coil, because it's in between the MR head coil and the TMS coil and the head. And so this coil here, developed by the Winnisberger lab in Vienna, is basically, it's not shown here, like a 7-channel surface RF coil, which is very thin, and it's then placed in between the TMS coil and the head. And you're stimulating through this imaging coil, but the imaging is not disturbed by the TMS coil because it's not in between. And you get very good SNR at the surface at least. So for cortical imaging, that's optimal. And they've shown in block designs, again, can map activation in the motor cortex, but mainly for superthreshold intensities. So, as I said, the problem is you stimulate the motor cortex, you get a muscle twitch, and then it goes the same way back, and you activate your cortex. So what can we do? We have to block that connection. And there's the method of the ischemic nerve block, which is also used for like topical, like anesthesia-like effects when you do a local hand surgery, for example. We use a very mild version of that. So we inflate a blood pressure cuff to the systolic blood pressure for about 25 minutes. Then SCPs are completely blocked, MEPs are completely blocked, and so we then have a time window of about 20 minutes where we can do stimulation of the motor cortex without getting MEPs and without getting any afferent feedback. So after roughly 50 minutes, we release it again, and then, well, there are a few bruises, a few petechia, right, but it goes away after 1 or 2 days. So here you can see stimulating at 120% of the RMT before and during the ischemic nerve block INB, and the MEP is like completely gone. That's our setup. So we stimulate the left primary motor cortex at different intensities. Here I'm focusing on the superthreshold intensity. We also measure MEPs in the MRI, which requires the hand to be fixated, because otherwise the electrodes move in the magnetic field, which gives you strong artifacts. But we were able to get good MEP measurements. And we first wanted to see whether with this special design, so basically we have 2 of these surface coils, one under the TMS coil and one on the other side. So 14 channels, which allows like whole brain coverage. But we wanted to make sure that we have good sensitivity, right? So we did a finger tapping task. And so with the setup and with the fMRI sequence we use, we get good measurements from the sensory-motor cortex, right? If you activate it spontaneously by moving your finger, this is what we get. So that's good. So we know that we can detect bolt responses from below the TMS coil. And then we did another check. So in one session, the participants came in and got stimulated electrically at the median nerve at the wrist to produce CMAPs, so compound motor action potentials, like basically maximally stimulating the median nerve, which is a mixed nerve, the afferent and efferent. So it produces motor twitches, and we adjusted them to be similar in size, like the MEP you produce, to the MEP we produce with 120% of RMT when we do TMS. And well, we have a pre-measurement, then we inflate weight, and then we do the peri-INB measurement. And so here we see sensory motor activation for the sensory stimulation, as you would expect. This is during the INB, same threshold. It's completely gone, and that's the difference. So just to show that, like for each subject, we showed that we can successfully block with this technique the input to the sensory motor cortex. And then we did another session where we went over the TMS coil, also showed that MEPs are blocked. So we know that afferent and efferent pathways are blocked. And again, before the INB, we see stimulation, we see activation to single TMS pulses in the sensory motor cortex. During the INB, it's mostly gone. There's a little bit left, and we're now doing surface-based analysis, which are more sensitive. So we still have to see whether like something is left or not. I'm not sure about that yet. But there's this very strong reduction. So the majority of the activity that you see on the TMS coil is not due to the transcranial stimulation. It's due to reafferent feedback. So that's very important for studies of the motor cortex, which are like 80% of the published studies in TMS fMRI. So it's mainly, but not entirely, caused by this referent feedback when you go for suprathreshold intensities. By the way, also a problem for TMS EEG, right? So of the motor cortex, when you stimulate above threshold, you will get feedback, referent feedback that's going to desynchronize your activity, and you will see alpha, beta desynchronization, all these effects. Now moving from cortical to subcortical regions as targets, which we can only target indirectly, right? And especially, for example, the subgeneral SEC for treatment of depression, also the ventromedial prefrontal cortex, they are very interesting regions we would like to stimulate, and we can't directly with TMS. Well, we might be able then with focused ultrasound, but for now to target it indirectly, we need to go via connected regions. And there's a lot of research based on the desire to optimize treatments for depression with RTMS, all the work by the group of Michael Fox and Robin Cash, showing that if you target a brain region in the DPFC that is maximally anticorrelated with the SGACC, you'll have optimal treatment outcome. So this is resting state function connectivity, and it helps you to personalize the target, and so basically the coil position. And like the distance between the actual coil position and the optimal one in terms of anticorrelation with the SGACC predicts treatment outcome quite well. And now the Stanford accelerated protocol for treatment of depression, which just received FDA approval recently, that's the same protocol. That's also using this kind of targeting procedure, right, to be specific for this functionally defined targets. And it's very effective. But there's still some things to optimize, because we're thinking about a single coordinate here, right? And it's just perpendicular scalp projections. The coil orientation is unclear, or just like done as it's always done for the DLPFC, right? But and then there are E-field simulation approaches to optimize the intensity for a certain target, right? So you can, basically with the brute force method, simulate for different coil positions and orientations the E-field, and then select the position that gives you the maximal E-field for your target coordinate. But again, it's like for a small region of interest. And we might have off-target effects, right? Because if you look at the anticorrelated cluster, as this orientation, well, here the E-field produced looks very different. So we are also stimulating a lot of even positively correlated regions. So we might need to stop thinking about our cortical targets with TMS as like small coordinates, right? We're stimulating regions based on the E-field. So what we did, or what Max Lückel, a PhD student in my lab did, is to then have a combined approach where we maximize the overlap between the induced E-field and the anticorrelated cluster, while minimizing the overlap of the E-field with a positively correlated cluster. So to maximize on-target and to minimize off-target stimulation, which results in very different coil orientations depending on the participant. And then we went to the scan again with TMS fMRI to validate it, right? To look at our proof of target engagement. So we have this VMPFC-based functional connectivity maps, do the E-field optimization. Oh yeah, I should mention, also select the intensity then based on the E-field model, like trying to get about 130 volt per meter in the like 5% of maximally correlated voxels, and then go for the validation. Not going into the details here. Here we use multi-band, multi-echo sequences, which are very sensitive, and so we actually work with a few single subjects done so far. So this is single subject level. And we actually got bolt response under the TMS coil, which looks very similar to actually the E-field. So we got a local activation where we have the maximal E-field. Didn't investigate that yet systematically. We will like to look into correlation between E-field and bolt response. But we've got a positive correlation here, and sorry, a positive bolt response, and a negative, basically a drop in the VMPFC. In another subject, again, the cluster we want to target, and again, we get an activation bolt response at that cluster, a negative bolt response in the deep brain regions like SG, SSC, VMPFC, part of the same network. You could argue, well, I mean, these regions are also involved in like pleasant, unpleasant stimulus processing. Maybe this entire procedure is just activating that. We don't have a sham, but there's something better. We have a very good control side. So we basically move the coil a little bit, optimizing it to hit the uncorrelated voxels, neither positive nor negative, but keeping coil orientation similar so that you would get similar click sounds, similar sensation, but we stimulate off target. And this is the contrast, the negative versus the neutral target. And you still get these deep brain activations, right? So that means you move it a bit, it feels almost exactly the same, but you don't get the deep target activation. So it seems that this method works for targeting not single structures, right? I mean, that's not the brain, how the brain works, but networks involving these deep brain structures, for example. And we're now investigating this in a larger sample, looking into stimulation of negative clusters, positive clusters, and the respective control conditions. Okay, so changing gears from fMRI to EEG. fMRI is great for whole brain coverage, great spatial resolution, and enables us to provide proof of target engagement in deep brain regions. But when we are interested in more quickly fluctuating brain states in oscillations, then we need a technique that has superior temporal resolution. And EEG is perfectly suited to study oscillations. MEG would be as well, but you can't put a TMS coil into an MEG system. It would blow up the system. Maybe with OPMs in the future, that might work. So why interested in oscillations? Well, they might define brain states, right? Most of these oscillations, like synchronized brain activity, is generated in some thalamocortical circuits, which are under influence of neuromodulators like acetylcholine, noradrenaline, etc. And when you go from different vigilance, from one vigilant state to another, from active wakefulness with some beta activity, or then you get relax or close your eyes, you have some alpha activity and get more drowsy, theta kicks in. During sleep, then sleep spindles are observed. During deep sleep, slow oscillations, strongly synchronized brain activity over the entire cortex. Well, hopefully you don't go into coma. There's like even more synchronized activity. And this amount of synchronization correlates with the level of consciousness, but that would be a different talk. Now, these oscillations are relevant for a lot of different cognitive and motor functions, like all over the place, and they are often disturbed in neurological or psychiatric disease. So why are oscillations like involved everywhere? I think because they are like basic building blocks of information processing in the brain. And the most fundamental function might be what I would call input-output gain modulation, right? So you have, independent of how these oscillations are precisely generated, like what are the neurophysiological mechanisms, they have more excitable and less excitable states. And so if during a more excitable state an action potential arrives in a certain neuron population, it's more likely that it's going to cause an action potential again. It's like signal transduction happens. If it's in a down state, like either actively inhibited or just less excitable, less background input, it's less likely that an incoming action potential is going to result in signal transmission and an outgoing action potential. Very basic, but very important, and it has an impact on many other higher level functions of oscillations. So just going quickly through it, you know about communication through coherence, right? So phase locking oscillations to make sure that the duty cycles, the upstates overlap, actually allows information transfer. If they would be out of sync, like these two, then information processing wouldn't work because like up and down state are overlapping. Cross-frequency phase amplitude coupling, same thing. You have slow oscillations with more and less excitable states, and during more excitable states, the amplitude of faster oscillations is increased, like here for delta theta gamma coupling, or very classical also for slow oscillation, spindle, ripple coupling during sleep. You have even more complex than phase precession, right? Theta gamma in the memory system, but maybe also alpha gamma when scanning visual scenes. And then another principle that I will come back to later is phase-dependent plasticity. So that maybe the arrival of fast oscillations in a state of increased excitability during a slow oscillatory state causes increased calcium influx into the dendrites, into the cortical neurons, which will facilitate long-term potentiation, while if the same fast oscillation would arrive during the down state, it might not produce sufficient calcium influx to produce LTP, or maybe even produce LTD instead. Now, how can we make use of that for brain state-dependent brain stimulation, right? So if we do not treat the brain as a black box, like a static thing that we just stimulate and then get our MEP outcome or whatever our outcome measure is, but if we see it as the dynamic system that it actually is, and we consider oscillatory states or functional connectivity states at the time of stimulation, we might get very different outcomes. And if we would know the state before we stimulate, then we could target specific states to get specific outcomes. Brief words about the terminology. So what we typically do is like open loop brain state independent stimulation, right? We just trigger at some point with a certain stimulus interval and then get an output measure. What I'm going to talk about today mainly, and what most of the studies in the field that are doing like real-time EEG trigger TMS are doing, are open loop brain state dependent brain stimulation. So we monitor a brain state like an oscillation, or analyze the EEG signal in real time, and then trigger the stimulator. It's open loop because in this case we might not modify the variable that we're monitoring. Right? A closed loop system does that. So like the radiator, right? If you have you measure room temperature, and if it's too cold, then there will be more hot water influx into the radiator, increasing radiation, increasing room temperature. And when the temperature is reached, it stops doing that. So that would be a closed loop. But so if we measure MEP amplitudes, for example, during different oscillatory phases, as I will show, without changing like the oscillation per se, then it's not closed loop. So it's mainly this what we do. You often read closed loop, but there's hardly any true closed loop non-invasive brain stimulation happening. How do we do that? So we've developed some technical help for these kind of experiments. My son, a PhD student in my lab, he developed the BEST toolbox. You might have heard of that, the Brain Electrophysiological Recording and Stimulation Toolbox, which allows open but also closed loop experiments. Closed loop in terms of, for example, threshold, closed loop automated motor threshold hunting, real-time experiments targeting different phase angles, doing input-output curves, doing paired pulse stimulation, RTMS, you name it. You can also trigger focused ultrasound stimulation, etc. So basically receives a lot of input from different hardware systems, either a bit more jittered, which is not real-time compatible, or if you have a real-time capable EEG system, then which is fast and consistent in streaming, then you can also use it for real-time phase targeting. And then we trigger different machines, whatever is able to be triggered by a TTL pulse, and we can set the different intensities or current direction or whatever. And parameter configuration is a bit slower. It depends on the machine that you are controlling, but the simulation is then done like with a very high precision, the triggering. You can design multi-session, multi-protocol experiments, set all the parameters, save it, share it, and define different hardware and output systems, get online results during the stimulation already to see whether data quality is good, and get some of the basic analysis even done like in real time. So if you're interested, have a look at bestmindstoolbox.org. There's a documentation of the technique, there's a help forum, and so we're always interested in helping you set it up for your lab, depending on the hardware that you're using. Now, following this advertisement, going back to the kind of closed loop or open loop EEG to TMS applications. And I'm using them because I have an interest in the new physiology behind oscillations. And we often have assumptions about what these oscillations are doing, but to test how their excitability profile is, like to characterize, or actually the excitability of the cortex is modulated by the oscillation, or at least is co-fluctuating with the oscillation. We can trigger TMS in real time with high precision according to the phase of specific oscillatory frequency bands. For example, we wondered whether the mu alpha, that's the alpha oscillation in the sensory-motor system, whether that behaves like the visual alpha, which is supposed to be inhibitory, right? So visual alpha is believed to be pulsed inhibition. So we wanted to test for the sensory-motor cortex, where we can measure MEPs and thereby assess excitability, whether we do have something like pulsed inhibition, that with increasing amplitude of the oscillation, we would have increasing bounds of inhibition, but always the same flow here where there's no inhibition. So where you would have the same excitability as if there would be like no oscillation when it's desynchronized. The other scenario would be the opposite, like the pulse facilitation. That means with increasing amplitude, you have phase angles where you have strongly facilitated excitability, but then still phase angles where it's the same level as if there would be no oscillation or very low amplitude oscillation. And then there's, of course, the possibility that it's just symmetric, like it looks in the EEG because you use AC filters, that there's facilitation inhibition. So we targeted explicitly the lower 20% and the upper 20% of the individual mu power distribution of a given subject. Did so either at low power, was basically desynchronized at random phase, because here phase would be just noise, right? Filtered noise. It also gives you a phase, but it's meaningless. But for the full-blown mu alpha oscillations in the left motor cortex, sensory motor, we then targeted 4 different phase angles: peak, falling flank, trough, rising flank. And we did so with single pulses to measure MEPs, and with paired pulse to measure short interval intercortical inhibition to see whether there's some GABAergic, GABA A receptor mediated inhibition that's modulated by phase. So just to show that the targeting worked, here the power spectra for single and paired pulse. For the low amplitude, like random desynchronized condition, there is no peak, but for all the other conditions, as it should be. And here are the time frequency representations. Time point 0 is when the TMS pulse happens. It's normalized, like Z normalized across all the conditions. This is why it looks bluish here. But you can see that it is restricted to the mu alpha band. It is also restricted to the left sensory motor cortex, because we use a local Laplacian montage to get the signal really from this region and not have like occipital alpha, which has much larger amplitude, spreading into our EEG electrodes at the motor cortex, and of course spurious phase angles that were not intended to measure. And then finally, of course, here the averages of the targeted trials. There's no phase during the desynchronized condition as it should be, but we target the peak, falling flank, trough, rising flank of the mu alpha oscillation. Here are the MEP results normalized, and the interesting thing is that, like relative to this random desynchronized state, we have no difference to peak and falling flank, but we have an increase for trough and rising. So that means there's no phase angle during the mu alpha oscillation where the excitability is lower than during the desynchronized state. So we do not have any absolute pulsed inhibition happening. We have only pulsed facilitation happening. In the motor cortex, we do not have any phasic modulation of short intercortical interval intercortical inhibition. So in another study, we now just targeted 10 different power bins, basically. So just looking at the different oscillatory amplitudes, independent of phase, and again, see normalized here, also just normalized power spectra. So we targeted really nicely different amplitudes, and you see, if anything, a weak positive correlation. The larger the mu alpha oscillation amplitude is, the larger the MEP. Not like in the visual cortex, the larger the alpha oscillation, the lower the likelihood to perceive a phosphene, like for example Gregor has shown, right? So this was not expected. This is why we did these two experiments to confirm. And actually, we just did another sample. So these two studies have been done during my time in Ulf Ziemann's lab in Tübingen. Now we acquired a completely new sample in Mainz, in my own lab, and compared it to not just lower 20%, but we used 1/f correction, the power spectrum, to look at states where there's no oscillation present, as defined by no oscillatory bump that goes beyond the power level of the 1/f background distribution. And again, trough and rising flank have the highest excitability. Here, the fall, in this case, the falling flank has the lowest. We also looked at different power intervals again, and I'm not so sure about the last one, so we're checking whether that's really true or whether there's some artifacts, but again, it's, if anything, a positive correlation, or maybe it's starting to U-shape in the end. So we could confirm that, pretty sure that it's true, but what does it mean? So it seems we have pulsed facilitation in the motor cortex, but it might actually be explained by most of the signal that we get, the EEG signal coming from the somatosensory cortex, from the crown, right, the radio source. This is where the mu alpha is generated. And it might actually rhythmically disinhibit the frontal cortex, because due to the tight connections there. We're currently analyzing a data set where we looked into short afferent inhibition and SCP modulation to figure out what's actually going on in this sensory-motor interaction, but I can't give you the results yet. Just so much short afferent inhibition seems to be clearly modulated by phase and amplitude as well. Okay, so we can use this technique to test specific neurophysiological hypotheses about the function of oscillations. And when we have a good reference, like the desynchronized state, we can even tell whether is it facilitation or inhibition that's happening. Colleagues from Tübingen they'd actually shown this principal difference between peaks and troughs even beforehand, and then repetitively targeted with 100 Hertz bursts of TMS, either in one session only the peaks, which are less excitable, or only the troughs, which are more excitable. When you do that for a while, then afterwards there's a LTP-like increase in excitability if you targeted the more excitable phase, but no such increase, or maybe trend towards a decrease if you target the less excitable state. So you can use it also to induce maybe phase-dependent plasticity. So this idea is inspired from sleep and slow oscillation, spindle coupling. It's also present like in theta-gamma coupling. There's work from Wert and Lismin, from the 90s, even showing that electric stimulation in the hippocampus targeting different theta phase angles to get LTP and LTD-like plasticity. So this phase-dependent stimulation with TMS may tap into the same mechanism as this cross-frequency phase-amplitude coupling. So talking about sleep, we did not only investigate these excitability changes during mu-alpha during wakefulness, but also during sleep. Actually, this is the first study with like EEG trigger TMS that had been done. I did this during my PhD in Hartwig Siebner's lab. We targeted the slow oscillation of deep non-REM sleep, either the up states here in red or the down states in blue. But this one was in this reclining chair, semi-upright position, and we measured MEPs. Had fixated, navigated, very comfortable, like noise on your ears. But so half of the participants managed to went into actual like deep slow wave sleep, and we could get a good number of events. We also looked at the TEPs here, in red, like when targeting the up states. So we also sometimes just detected them, but only set a marker and did not do TMS. That's important because, as you can see, well, when you do your time-locked average according to a targeted oscillatory phase, then you will get, of course, the average oscillation as your background activity. Right? Oscillations only average out if they're not phase-locked to the stimulation, and here they are. So this is how the oscillatory events look like that we're targeting, and here you can see the extra TMS-induced activity on top. If you subtract it, this is what you get. This is how TPs typically look like during sleep, as here compared to wakefulness. So it's a stimulating the same region, but now instead of a short complex event for 300 milliseconds, you get like 1.5 second long, full-blown, slow oscillation-like responses. And Marcello Massimini has done several studies and shown that. So what we showed here for the motor cortex is that not only compared to wakefulness, there's a decrease in excitability, which can be partially explained also by spinal inhibition, but there's a consistent difference between up and down state, actually for every single subject. So it's a very consistent thing. And also the TMS-evoked response, while looking very similar for up and down state, there's a difference in the early at the stimulation site. It's larger when you stimulate during the up state than during the down state. And then also the spreading oscillation over the entire cortex is larger when stimulating during the up than during the down state. Single trial correlations between slow oscillation amplitude that we stimulated and MEP showed like a correlation restricted to the left sensomotor cortex and the, well, you can see a little bit in the contralateral homologue, but not significant. So it's not an all or none phenomenon. It's the voltage of the oscillation, of the desynchronization, the depolarization or hyperpolarization associated with up and down states, that is, the amount is predicting the MEP amplitude. And for the EEG, for the TAPs, of course, we couldn't do like single trial analysis, but we binned it and looked at low, middle, and high amplitude slow oscillations that we targeted, like post hoc determining this, and then here the TPs again also differ, right? I mean, if you target a large oscillation, then you also like a strongly depolarized state, you also get a large TP response that looks again like a slow oscillation. Because that's basically what the cortex can produce during deep sleeps in a bistable state. We stimulate the cortex or TMS with some kind of other stimulation, it produces this kind of effect. And now recently, like 10 years later, we went from slow oscillations to spindles. Again, Omer with a setup, now it's not a reclining chair, it's a bed. We do this also new navigated. We use vacuum cushions to stabilize the head. At the moment, we can do naps of a few hours. We hope in the future to be at some point able to do whole nights. And now we wanted to investigate what's happening during the sleep spindle. Right? Sleep spindles are very important for memory consolidation, memory reactivation. And we have shown, for example, with intracranial recordings in epilepsy patients that in the hippocampus, the sleep spindles also group hippocampal sharp wave ripple events in their troughs, right? So for slow oscillation spindle ripple coupling, this cross-frequency organization is very important to allow then a fine-grained hippocampal neocortical dialogue. We still don't know what the spindle does actually in the cortex. Does it like alpha produce a rhythmic inhibition or facilitation? And for the spindles, it's because they happen rarely, you really need an EEG triggered system because otherwise you would just stimulate all over the place and hardly hit any spindle. So we wanted to stimulate during spindle-free and also slow oscillation K-complex-free intervals as a baseline again, during non-REM sleep, and then during spindles targeting rising flank, peak, falling flank, and trough. And then also later during the so-called refractory period, like 1.5 seconds after the spindle peak, this is the time we typically do not get another spindle. So we wondered whether there's some kind of inhibition going on or something. So to do this, first, a real-time spindle detection algorithm had to be developed. That's what UMA did. So it uses a lot of derived signals in time and time frequency space to ensure that we are detecting spindles. We can't detect them in the beginning because we don't know whether they're going to grow, right? But we can detect them pretty close to the center of the spindle. And actually, compared to offline, so established offline detection algorithms, we get very good alignment with those. So same accuracy, actually similar to how good those algorithms, these offline algorithms that know the future of the spindle, right? Not only the past as we do, as good as they are corresponding to each other. You never have perfect correspondence, neither between human raters nor between algorithms. But it works pretty well. So we can target the peak, falling flank, trough, and rising flank again of the sleep spindle. As you can see here also in the time-locked averages, you see the spindle happening here. We also ensure it's not driven by any other neighboring oscillatory band, because if you would have an artifact in the EEG and then you have a bandpass filter, then it looks like the spindle is happening, but then you would see something in the neighboring bands, and we ensure that that's not the case online. And so we use the C3 against contralateral mastoid, typical sleep scoring montage. And we actually, the spindles we detect are also a bit localized to the left sensory-motor cortex. Just a quick figure showing here the spindles of the different phase angles, and then afterwards the TMS evoked potential, which we have not yet analyzed in detail whether there are any differences, but we were interested in the MEP modulation by the spindle. And this is what we get. There's a clear modulation again of MEP amplitude by the spindle. It is like for alpha, mu alpha, it is largest during the trough, and it's smallest during the falling flank. But the baseline is different. So the spindle-free intervals and also the refractory period, which didn't make any difference, they are basically at the same level as the trough. So we have asymmetric pulsed inhibition during the spindle, while during wakefulness in the mu alpha, same region, we have pulsed facilitation. So if we put all these results from slow oscillations and spindles during sleep and mu alpha during wakefulness on one slide, then you can see that, that nicely replicates for wake mu alpha. It looks very similar, actually, for the spindle, but the reference is completely different. So we don't know whether that depends just on the background state, because we have a different baseline, basically, from which the change happens, but still the trough is the most excitable. It just doesn't differ from background state. Or whether it's really a completely different mechanism, right? To what degree spindles and alpha in the sensory-motor system are generated by similar mechanisms is like a 30-year-old debate. Most likely both are influenced by the thalamus, but for sleep spindles we know pretty well how they're generated. For alpha, not as well, right? But thalamocortical loops definitely play a role in both. So why is this interesting in the first place? Not only that we know that actually excitability is modulated during spindles, but we can now use that information and target most excitable states repetitively during sleep to induce plastic changes in the sleeping motor cortex, which could have long-term implications for therapeutic stimulation, right? Using this underutilized 8-hour window of sleep in patients, for example. And also we know that during spindles, reactivation of memory traces happens. At least we have a lot of data supporting that. But so far we can't tell whether it's causally, whether they're causally necessary for memory reactivation and memory consolidation. So when we can target spindles and disturb reactivation processes, we can make a causal claim that spindles are required for this memory consolidation process. And these are the projects that are going to follow in the next years. So what else is supposed to follow? Well, we would like to close the loop at some point. What are the problems? So, I mean, we can do all the real-time analysis, right? That works. We can trigger the stimulator in time, and we can stimulate the brain regions we're interested in. But here are the problems. Challenge one would be the real-time extraction of target brain states. So far, I've been talking about amplitude and phase of local oscillations. That's very basic, right? I think you will hear something also about functional connectivity estimates in real time, maybe from Paolo later. But if you think about more global, complex brain states, maybe using artificial intelligence or more derived complex measures that you can estimate in real time, that's always important, right? We could target more meaningful brain states for certain cognitive functions. Then, real-time removal of stimulation artifacts and confounds, right? Because when we stimulate, we get a lot of artifacts in the EEG. I could talk for hours about these artifacts, and I think many of you as well. And so this makes us blind for a while. So we can't target each oscillatory cycle with TMS if we may change those cycles because we have all the artifacts, and the removal offline is already difficult, online even more so. And then finally, even if we are able to detect complex brain states and get rid of all the stimulation artifacts, how do we actually control brain states by stimulation, right? I mean, it's one thing to stimulate during a particular phase and measure an MEP. It's a different thing to shift a complex brain state towards a desired state into a different attractive state, right? How does that work? And there, I guess we need help from control theory, etc., to get good ideas about when and where and how do we need to stimulate to actually change brain activity, right? Because only then we can close the loop and move explicitly the brain towards the desired brain state or away from a dysfunctional pathological state. So I hope this is where we're going to go. I would like to thank my group in Mainz, my collaborators and funders, and I would also like to announce that we have a few postdoc positions available, one actually on closed loop or EEG to get TMS, and also two on combined focused ultrasound with MRI in humans, another one for some focused ultrasound for blood-brain barrier opening and targeted drug delivery in mice, also within a collaboration. I'm not doing mice myself. So if you're interested, you will also find the job descriptions on our website. So thanks a lot for your attention, and happy to take some questions.
Thank you very much, Til. Are there questions? No questions, so I'll break the ice. About your first experiment that you presented on the no activation of M1 when you combine TMS and fMRI. So I was wondering if you would expect not to find any evoked potential after the TMS, coming from the direct stimulation of M1, if you were doing TMS EEG co-registration instead. Because we know that fMRI and EEG record different signals, right? So they capture different processes in the communication between neurons. So will you also expect not to find M1 activation in TMS EEG?
No, so I would expect to find motor cortical EEG responses when stimulating motor cortex. And also during ischemic nerve blocks, I would still expect to find them. But it's actually, it would be a very nice experiment to see which of the components of the TEP evoked at the motor cortex are due to reafferent feedback. Because, I mean, typically the early components we look at are, like, if you think about N45, P30, or at least N45, it's enough time for the signal to run down and up again. And it would be interesting, like, which components might disappear when you do this ischemic nerve block. I just don't think that I can get anyone in my lab to use this technique again, which is really difficult is finding subjects after a while, because it's not super pleasant, this technique, right? I mean, if you raise your arm for a while and drain the blood and use low pressure and then move the arm afterwards, and there are a lot of tweaks to make it less uncomfortable, but it's nothing we would now use as a standard technique in our lab. But it would be really interesting to see, actually. I think there are a lot of other TP components that are more explained, but thalamocortical or cortical-cortical interactions, and they should be preserved, right? But the part that might be due to reference feedback, yeah, would be interesting. So I'm happy to share information about the methods, and maybe someone is going to pick this up. Yeah, it's a good idea. Yeah, Walter.
Time is very relevant.
Yeah, yeah, right. I mean, of course, there's one caveat. If you have a like deafferented cortex for a while, I mean, it's not completely different, but just this is blocked. It might change the excitability as well, right? So it could have some implications that are more complex than just removing this afferent input. But that's the best method we could think of.
So very impressive data. The question to the motor cortex stimulation. So what I understand, the location is confirmed in the wall. However, you say, when you showed early in your talk the data where the motor cortex was activated by TMS in the fMRI.
Yeah.
So it was really confirmed that it is in the wall. However, a substantial or large proportion is reafferent induced. This is the cosine. So what we see here is it's deep in the sulcus, the activation.
Yeah, so I mean, for both, I think finger tapping evoke both responses.
Yeah.
That's also typically in the sulcus, right? And here it seems also to be, yeah, it's a bit hard to tell.
So this is tapping, not TMS.
No, this is TMS, but I mean, yeah. We also know for tapping that it's always deeper, yeah.
Yeah, what is particularly interesting is that obviously deep in the sulcus we have a hot spot, and the question always is, if you increase the TMS intensity, you get a latency jump to shorter latencies. That may have two causes. One is that in the layer 4A, you stimulate first with a lower intensity the axon hill, and then if you increase it, it jumps to the bend. The second explanation would be that if you go to higher intensities, you target now area 4P deeper. And this looks as if the area 4P with the large monosynaptic connections to the spinal cord is targeted.
So that's interesting. I mean, so we're at the moment doing more fine-grained analysis at the surface, like in surface space, where we can look really into like differentiating sulcal and gyral activations in a bit more detail. We also have stimulation at 100% MSO, at 100% resting motor threshold and 120%. So we could actually compare these two intensities and look at whether they activate different parts, not only like different levels of activation, but whether the localization changes systematically with stimulation intensity. That's actually an interesting thought. Yeah. Could look at that. Yeah. Yeah, it's hard to say from these. I mean, yeah, so next time I'm going to present surface-based analysis, and then it's a bit more clear. So this is still preliminary, right? But it's actually an interesting question that we could tackle with this information. Also, another analysis that we haven't done yet, or at least on the first quick shot, nothing came out of it, is using the MEPs that we have acquired and use them as a parametric modulator, basically looking for whether MEP amplitude predicts bolt signal. And so far, interestingly, it didn't. It didn't predict it, neither for the 120% nor for the 100%. But we have to do a few additional analyses to be sure that there's really nothing. But I would have expected larger MEPs to produce either stronger reaffirm feedback, or maybe larger MEPs being indicative of higher excitability, and then therefore stronger responses in the cortex. So I mean, we did all this effort to record the MEPs to model this, and then it doesn't do anything. So I'm not yet convinced it doesn't do anything, but so there are a few more interesting analysis to come in the next few months, hopefully. Good. Yeah.
Thank you for this very interesting presentation. I was wondering, you mentioned that continuous closed-loop stimulation is not feasible, and I've seen some very interesting low-intensity TMS posters here at this conference. Would it be feasible if we just reduce the TMS amplitude?
Yeah, good point. So I wouldn't say it's not possible, it's just really, really difficult, and so far it hasn't been done convincingly. At low intensities, I think, I mean, some of the artifacts will disappear. You might not get any cranial muscle responses anymore. You might not get strong motion artifacts and decay artifacts. But you would still have pulse artifacts in the EEG, which are still larger than your true brain activity, right? And one has to be very careful when removing those artifacts that there are not any residual artifacts, any discontinuities introduced by the cutting, interpolating, etc. So it's really hard to show this, these entrainment effects, I think, also for low-intensity pulses, because one is doing a rhythmic manipulation of the data, right, due to the also post-processing, which can easily result in some spurious rhythmic activation. So in principle, I think that low-intensity TMS subthreshold is able, should be able to entrain brain activity. So at least at levels, for example, like 60% resting motor threshold, because we know that you can get still short intercortical inhibition. So 60-70% does something to neurons, to inhibitory neurons, which are also important for generating oscillations. So I think that works. With the very, very low intensities, also have an effect. I don't know. Right?
I'm asking because not with a view to entrainment, but with a view to sort of enhancement and suppression. Because if you have continuous closed loop, you have a sort of a different way of interacting with the oscillation.
Yeah. So I guess when you would, that's true. So when you would really for longer trains, maybe even for minutes or so, right, hit each cycle with a low intensity pulse. Basically, same idea as with TACS, right? You have very low intensities that might not do anything per se, but they can slightly bias then activity in one or the other direction, and then over time you might be able to see an increase or decrease depending on the phase you target, right? Yeah, I think that's definitely something that would be testable, right? Closed loop, low intensity for longer time periods. Yeah. I have some hopes for focused ultrasound in that regard as well, right? Because turns out it's not that you don't get any artifacts because you have these piezo elements and electronics and stuff, but at least you don't have these electromagnetic fields that interact, right?
Are you currently working on closed loop focused ultrasound, or?
Not currently, but planning to.
Thank you.
Could be part of the advertised postdoc position, actually.
I may just add to this question, Joel Tour in our lab published a few years ago that you see with phase locking value analysis effects in the EEG by a voltage of 35 volt per meter, and it just failed with 20 volt per meter. But I'm sure if you had a larger number of subjects, you see, of course, the influence in the EEG with low intensity TMS.
But also the phase locking is also sensitive to rhythmic residual artifacts or small discontinuities in the data, right? So it's really hard to nail this down.
I have to say we had a double acoustic blinding, so earplugs and then noise cancellation phone above that. So we had a lot of effort to avoid that. But I agree, this is a really tricky thing.
Yeah. Okay. Otherwise, lunch?
Yes, that's time for lunch.
Thank you.